thank you for the organizers for wrapping up this amazing event, for inviting me here, and I will comment on the impact that this symposium will probably have, and hopefully will have in my last slide. So, I would like to present not really results, but perspective on the research that got funded very recently by the INR, which is a funding agency in France. It's very competitive. And the title of the project is the Valorization of antimalarial Artemisia in the South. South means southern hemispheres, or in countries where malaria is really a plague, countries with low income, developing countries. And the idea is really to bring scientific solutions to evolve those antimalarial Artemisia into standardized phytomedicines and possibly source of new drugs. Next slide, please. So, the context of the project, I will probably start with the therapeutical context. The situation is pretty alarming because we have the frontline drugs that are falling. If you look at the main enemy, which is Plasmodium falciparum, which is the killer species responsible for most malaria deaths, well, you have resistance that arose following artemisinin-based therapies in Southeast Asia, in the Greater Mekong region, some 20 years ago. It's now prevalent there. It's the only dominant that you find there is mutant in the K13 propeller gene of the parasites. It made artemisinin-based therapy fail, and also the drug partner of artemisinin derivative in this biotherapy are also failing because of that mutation. And very recently in Eastern Africa, so around here, the same mutation appeared. So it's stable for now, but the situation can change very quickly. And it's probably metastable. And if ACT fall, just like chloroquine fell to resistance, we could go from the 600 deaths per year back to more than 1 million deaths per year. Problem is we have no new drugs to counteract a resistant Plasmodium falciparum. When you look at the other enemies, Plasmodium vivax, and typically the dormant form, it's relapsing species, and here again you're limited by 8 aminoquinolines, just like primaquine and tafenoquine. But there are limitations. They're dependent on the host genetics to elicit activity without toxicity, and they're kind of tricky molecules, and they have unique modes of action, and we have no better drugs so far. In addition, Plasmodium falciparum, if you want to block it, especially the resistant mutant, you need to treat gametocytes, which are notoriously drug resistant. And here again, you only have 8 aminoquinolines to target those stages. So, in this very context, next slide please, but also due to cultural reasons and due to limitation in the drug availability, to their cost, to their counterfeiting, which is very common in countries of endemicity, somehow toxicity, like I said, there was a massive return or new uses of Artemisia annua and afra, the one species that contains artemisinin and the other that contains very little or no artemisinin. So against those uses, you have the WHO, you have many pharmaceutical industries, you have many Western medical doctors. Like Metodius said, they are seeing annua as really unreliable because artemisinin fluctuating in concentration could lead to a therapeutic failure. They're seeing Artemisia annua as a monotherapy, like just seeing artemisinin, just like the tree hiding the forest. And we know the forest is just behind. And they say afra has no artemisinin, so it doesn't make any sense. For the use of those plants, you have many NGOs, you have the local population, trade practitioners, many scientists. And we are aware of the complexity of the plants, so we see those as potentially polypharmacological therapies. And in addition to a synergistic effect regarding curing malaria, there is also a very important aspect that if you have polypharmacology, resistance is probably going to be a non-problem because resistance would mean mutation, and the probability of the parasite making several mutations at a time to resist many molecules doesn't exist. So we also know that the uses are pretty ancient. For Annua is millennia, for Annua is more recent decades. And those differences in view were feeding also really north-south controversies, bringing back, questions about pharmaceutical monopoly, post-colonial intervention. So around science and lack of data, there is a lot of controversies because of opponents in cultures and views the way people should be able to treat themselves or not. Next slide. So those are, I won't say personal because I didn't make the experiments, but I participated to the ones made in Dominique Mazier's lab. We showed for the first time that the infusions were extremely active against hepatic stages of several species and several stages of hepatic Plasmodium species, including the relapsing one like Plasmodium vivax with its hypnozoites. So it was really striking to see that even Annua had really potent effects. And we also reviewed most of the known phytochemistry of the infusions, not organic extracts. It's very important because we were looking at really traditional use of the plant. And actually there are cocktails of compounds. And when you classify the compounds against their phytochemistry classes and modes of action, you can clearly see that monoterpenes and sesquiterpenes are known, some papers show that, to inhibit the prenatal synthesis enzyme of Plasmodium. That flavonoids are very good binders to heme, so they prevent, to hemozoin, they prevent heme detoxification by the parasite. You've got many molecules that create oxidative stress, generating ROS, reactive oxygen species, and those are keys typically to kill gametocytes and hypnozoites. So we have probably a number of compounds that are already telling us that they are targeting different stages. So it's really, really interesting. And it was also published in a French version to make availability more broad. Next slide. So, despite those positive results on our side, I didn't put all the reference because I would need probably several slides to put them all. But to put it in a nutshell, like we said in French, a female dog couldn't be able to find its puppies. It's really contradictory. And if you look at the typical Artemisia annua versus asexual blood stages of Plasmodium falciparum, which is the causative stages of the physiopathology of malaria, there is no correlation between data. Some argue and show very, very diligently that art concentration and activity are typically correlated positively, that artemisinin holds almost all the activity. And in other papers, you have the total contradiction, like infusions are almost 10 times more active than art alone when you standardize the concentration. Like you know exactly which concentration of art you have in your infusion, so you're allowed to really compare IC50 between the plant extract and artemisinin pure. So there is also data by Pamela Weathers on delayed resistance in an in vivo model of Plasmodium chabodi that was really striking. There is a delayed resistance compared to artemisinin alone. So it's really, really interesting. On liver and gametocytes, we don't have many data. On liver stage, I showed you the data. It's typically an art-independent inhibition. Artemisinin is active at nanomolar concentration on blood stages. It's acting at micromolar, 1,000 times less, 1,000 times more, sorry, on liver stages. Regarding gametocytes, the picture is really intermediate. There is typically artemisinin powerful effect on young gametocytes, but it probably fails away and becomes art independent on late stage gametocytes. For AFRA, it's also very cloudy, depending on the strains probably. Like I will try to convince you. AFRA is most of the time inactive on blood stages because of lack of artemisinin probably. And some other authors describe it as fairly active against those stages. On liver stages, we found very important activity, but we know it's art independent, so no surprise. And regarding the clinical trials, because this is what we all need to be looking at, the past clinical trials were showing beneficial effects and then recrudescence. Some were retracted due to pretended methodological weaknesses. I'm really not an expert, so I will not comment on that. You have one starting in Africa with a Boehringer industry under the guidance of Antoine Berry in Toulouse. Liver stages, there is, to my knowledge, no specific clinical trial. And on gametoside, there is a starting one by Agnès Louis, also in Toulouse, funded by a small fund. So she had a very small sample, but I think statistically speaking, she will just look at gametoside carriage. It's a very specific question, and she didn't need such a big N in the matter. So, better? Better?
Maybe. Just say.
Okay. So, my main hypothesis driving the project I will present to you is that the high heterogeneity of the samples that were tested are probably the main explanation for such inconsistencies in the activity, in the literature, and it's at least according to three variables: genetics, cultivation, and storage condition. Fourth condition would probably be season of harvest, like Mr. Luce showed, time of growth when you harvest. I think we all agree that most of the time it's made just right before blooming, so it's probably more homogeneous than we believe. The way you cut your plants or not after drying also is important, but at least we cannot look at everything. So let's say that genes, cultivation are probably first position. Maybe cultivation is even more important in terms of conditions, changing the chemistry than genes. We don't know that. And we know that because of those parameters, there is a highly variable phytochemical composition. It has been demonstrated in several papers. And of course, if you have variable phytochemical composition, you will have variable biological pattern. It is just obvious, I think, for everybody. So this is my hypothesis. And the only way I've found to try to answer the problem, to address it, is to really propose... I hope I don't get killed after the show. So the idea is to propose a systematic study embracing many lots of the two species worldwide with a focus on malaria endemic countries. And so if we analyze them properly and if we analyze them chemistry and biologically, we could probably try to see a clearer picture. And the general objective of the project is to establish phytochemical composition versus bioactivity relationship. And behind that, I have several scientific and social objectives. The idea is to give value to the plants, so we need to be hopefully defining active fingerprints, which are semi-quantitative mixtures of components, like between 0.1 and 0.5% of molecule number 1, and between 2 and 5% of molecule 3. And this would be also very easy to test, because most of the time those molecules are very simple. You can buy them. So after we have ideas of combination showed by the plant content, maybe we can synthesize those mixtures artificially and test them to validate hypotheses. The idea, of course, is to define optimal chemotypes that, for instance, Maison de l'Artemisia could use in a more focused way to bring those plants into standardized phytomedicines that probably would be better suited for clinical trials. We can also look precisely at minor compounds by mass spectrometry and see if some strains contain more than others. Maybe look for novel active principles that passed under the radar so far, and maybe they're very active, very low content, very hard to pursue. Maybe we can find novel platform molecules. Platform molecule is a molecule that can be used in bioeconomy processes, industries. And for instance, the small scheme on the right shows you that annua is containing dihydroartemisinic acid that is turned, artemisinic acid that is turned by chemistry into artemisinin via a dihydroartemisinic acid by a light process, just like Vitalius was saying. And this is the source of the drugs, ACT, artemisinin combination therapy. You can also make this precursor by yeast engineering. So these molecules can be seen as drugs, they can be seen as platform molecules. There is a lot of interesting chemistry in these plants, and maybe we didn't see it. The final idea is to propose good scientific solutions to bring socio-economical and therapeutical benefits to malaria plague population. And the societal impact we'd like to also have with this study is to try to alleviate or remediate those controversy regarding usage conflict, and to bring results that would be non-ambiguous because of their high end number, high coverage. Hopefully they will be better received by everybody. Next slide.
Next slide.
Thank you. So the project was funded this year. It will start only in March because I need time to really put everything together regarding resources, mainly plant resources. So we'll go from chemical and biological metabolomics with the VPN. Guillaume Marty is one of the leading researchers in France in modern metabolomics. So it's based on LC-MS analysis and also correlated to biological testing. And biological testing will be of two questions. I think it's very right.
Yeah.
Back to the old one. No? I don't know.
Yeah.
So we cannot study all parasite stages. Artemisia are very complex. I only look at malaria, I don't look at TB or HIV. And in malaria, it's complex again because you can look at dermatocytes, liver stage, blood stages, sky zones, hypnocytes, and it's just looking at the human stages. If you look at mosquitoes, it's probably another food story. So we'll just look at parasite blood stages, Plasmodium falciparum. We look at parasite liver stages, Plasmodium berghii as a model, Plasmodium falciparum, and hopefully the vivax entomology as a model of vivax for relapsing stages. And I'm just coordinating, so if you maybe think it's just, oh, it's not going to do experiments, it's probably the hardest. It's just try to make that work. But it's exciting. Thank you. Next slide. So this is the same slide in a more scientific way. So the first work package is really metabolomic analysis with a lot of LCMS trying to correlate activity to chemical composition. And activity will come from one work package on blood stage, the other on liver stages, and we will fuel WP1 with data, biological data from those two and three work packages. The algorithm that will process all those data through multivariate statistics will be fed by data such as inhibition at cutoff values. So cutoff is not an IC50. Cutoff, you arbitrarily fix a threshold of concentration and you see how much percentage of inhibition there are. So it also allows you to classify all the non-active plants and you tell the algorithm this is not active. We don't study them further. If the plants are sufficiently active, we will measure IC50, IC90, and IC99. And this will be done on lyophilizates from the infusion. We will fractionate the lyophilizate into 10 standardized fractions. And we also test the combination of compounds, just like I told you before. Based on the composition of active plants, we will synthesize artificially and test the mixtures. So hopefully, this analysis with big coverage will give us clues regarding active fingerprints, so combination of maybe not so active compounds alone, but very active when together. Paving the way to standardize phytomedicine, like which clone of Artemisia annua or afra have that sort of perfect mixture that makes them so active. And we look further deep into chemistry for possibly novel active principle and platform molecules for, fueling also industrial processes for interesting precursors, just like I showed. Next slide. So, the main of the talk is the biggest problem I have right now. And I think many of you are aware of it already because I've been talking to several of you. And this is really the basis of the project. If you want to construct and build a skyscraper, we need a perfect foundation. Otherwise, it will collapse. And if it collapses soon, maybe you have a chance to get out of it. If it collapses after you've been building the second and third floor because the basis, the work, the groundwork isn't good enough, it's time and money wasted, and it's also disturbing the cause. So how do we choose our samples? 15 is a good enough number, and if you go from in vitro to in vivo models of malaria, it's expensive enough. We have 800,000 euros. Seems like a lot of money, but it goes very far for such systematic study. So, first plan would be lazy, do a lousy selection, take whatever sample your friends or colleagues give you. Just bring 15 samples of one species and the other. Well, you could bring them into the algorithm, extract them, purify them, test them, it's fine. You will have very interesting results regarding composition to bioactivity relationship. That would be very nice. You could probably even say, okay, I need this compound with this compound with this compound. But it will probably not have further development because there will be no rationale. You will not be able to say, oh, this plant cultivated elsewhere will give the same. I don't know. Or maybe another clone cultivated in the same condition could do the same. You couldn't answer that. So behind science, there will be probably nothing back to the societies. No more development. It's not going back to the people. It will not have any other value than publishing, which is not my final objective, honestly, here. So we need a better plan. The other plan, next slide please, it's almost like ideal situation. I'm going to control those three critical variables: genetics, cultivation, storage. And I need to be pairing them so I can always compare samples. In science, you never ask two questions at the same time. You need to ask one question at a time, and you always need to have samples that can be paired so that you know where you're going. Otherwise, you will not very quickly. So instead of 15, you could have 16, having, for instance, 2 strains of anua in 5 countries, and decide to investigate storage, which is very important. Some lots are stable over time, very stable. Some aren't, and it's really lot dependent. It means that the composition of the plants impacts on the overall stability of the metabolites, which is very crazy and very tricky.
No.
Not yet. So that would be ideal. And for that, I said, okay, I'm going to rely on the Maison de l'Artemisia because they cultivate plants for so many years. They are probably the best people to know how to translate interesting plants to the people, to process them. They are really very important local actors at an international level. But of course, I will select malaria eligible countries. Like, I'm not going to study necessarily plants grown in Europe, even though they're interesting. I don't have resources to do that. So that would be probably ideal in this way, correlating science to future developments, because we will know exactly the relationship between climate. When I say country, it means country, you can take it as cultivation condition, like in Cameroon, plants grown together, or here in Tanzania, plants grown together. This is the countries are meant of controlling the conditions because plants are not growing so far away. So that would be probably ideal as a picture, but it's not possible. And I will tell you why in a second. So if I rely, for instance, on the Maison de l'Artemisia, there is a huge potential regarding diversity, but I will not be able to control everything. And so I will probably rely partly on the Maison de l'Artemisia for certain things, and it needs to be clarified, and probably the picture is not as dark as I say, but I will probably be a bit limited as far as I know things right now. If you look at Artemisia annua, Lucille Cornet started with 5 starting strains, and they were propagated non-clonally. So it means that in theory there is a genetic scrambling. But as a private information not so long ago, he showed me the plants in the field that also, you have the cabbage phenotype, you have the pine tree phenotype, and you have the oak tree phenotype. And it's true that there are only 3 phenotypes. So there are seeds, so they are probably scrambled genetically, but they don't seem to be that scrambled. And she told me also that in other countries, you got the same three phenotypes. So it's good for me to know, and I was really happily surprised because it means that there is probably not as much scrambling as I fear there is. And we need to control that. So instead of mixing the three, I would probably say, great, I have cabbages and oaks and pines grown together. Are they really related, chemically speaking, or not? And it will be very interesting. And which bioactivity do they have? Same, drastically different. So it's very, very interesting. For Afra, the situation is a little better, or at least calmer for me. 3 starting strains, and fortunately, Afra, because it's a perennial, it's propagated by cuttings. In some countries, the 3 starting strains are mixed up, so you cannot say this one come from there. I don't think they look very different, so it's tricky. But fortunately, in 8 countries, you have a monoclonal culture, and typically this is the Malawi strain that dominates, and you have 7 countries with pure Malawi strains. So it's really, really great. Maybe there are more. We need to discuss that. So it's limited as far as genetic representation, but it's already a big cluster of homogeneity for the project. So as a conclusion, almost conclusion, I think, but maybe I'm wrong, so you need to tell me if you have a better idea. I think I will try to take the best of 2 worlds. I will try to focus on strain homogeneous clusters and country homogeneous clusters. The former are defined by one strain cultivated in several countries. The later clusters are defined by one country with several strains. And this is very interesting because if you translate those words, it means that I could probably access cultivated samples on one side and field samples, I won't say wild type, but you know where I'm going, like really bush plants on the other side. And I think it's also important to have the idea that maybe plants growing in the bush are the ones we should be probably growing in the next 10 years. We shouldn't be limiting ourselves to what we have already. We need to take a mixture of both worlds, present and future. So this is a very silly clustering depiction. I'm not a bioinformatician by no way of whatever. It's just to give you the idea. I could have, for instance, for Afra, by the kind help of the FDA, a very homogeneous cluster of 7, samples, 7 countries with one strain. That would be very nice. And on the other version, I would have, for instance, with Methodius' help, via Professor Michel Friedrich, whom I've been talking to already, contact in Cameroon to access several strains of Anua, several strains of Afra, grown virtually in same condition, but we might control that. Same goes via Professor Ntumi in Republic of Congo, and I hope you help me gather more local clusters. I see many African researchers, many African scientists here. If you or your colleagues have access to locally harvested or locally grown plants, and you want to be part of that study, please step forward, because I need you to make this perspective more complete. Maybe not all will be prospected, but I promise that we will do at least phytochemical analysis. If we have many samples, more than 30 that I need, we will just do HPLC to say, oh, this profile is very much alike than the other. So we will pre-segregate redundant profile, we'll exclude them, and we'll only take the 30 profiles that have more diversity, and we can use them, probably. At the basis of that, we need to have genetic control. Is Anua really Anua? Is Afra really Afra? Sometimes you have contamination, so it can happen, and maybe we can dig deeper into variation, like if you look at metabolites, they come from biosynthetic clusters, and we can sequence them. So we can also prove that there are proxies, as far as genes are concerned, that are correlated to phytochemical composition. Next slide. This is the conclusion. So, it's very ambitious. It's more ambitious than my usual project because there is not just science. There are really a will to solve or try to solve a very complicated question on the Artemisia and to bring the community with solid deliverables. So you understood that understanding, controlling, and giving value to the chemistry of this antimalarial Artemisia is really, really challenging due to their diversity in the broad sense of the word, like genetic, cultivation, and many other parameters maybe. So we need high coverage and consistency, and we also need cutting-edge tech, like metabolomics. We need various but well-controlled plant resources, and like I said before to Dominique, and she smiled, I really want to know if behind this very inconsistent, very non-reproducible data, scattered plots, is there a Mona Lisa portrait in plants? I really want to see if there is. If there is not, we'll be back to trying our best, but if there is, we need to know it. We really do. So we need a lot of people. We have them in chemistry, biology, chemoinformatics. We need expertise from your kind of crowd, your kind of audience regarding agronomy. We need local actors. We need knowledge regarding drying. We need homogeneous procedures. We need good practices. And we need the MDA. And hopefully we can give in return the MDA key information regarding this strain is too toxic on our systems, on our models. Maybe petite aside, this one is very active, it's non-toxic. This strain has perfect combination and consistency all throughout Africa. You can grow it in Sahelian region. Chemistry is stable. I wish one day we could say that, we could find that. And finally, the project will be successful only if different people gather around common objectives. And this is my final word. I think the power of this symposium is to bring people that don't know yet, maybe, but we all do have the common objective is to bring this research ahead. And we need to share expertise, we need to share resources, knowledge, and plant material. This is really the foundation, like I said, because if we start working together on homogeneous samples, what we produce will be much higher quality research than working each of us on different strains, and one says active, the other says not active, and the pharmaceutical industries are just waiting for that to happen to give us the final shot in the head. Thank you.
Well, Romain, not bad.
I'm aging.
So I suppose that there is some questions. Or maybe not, but
I have just one.
Thank you so much, hard job. I have just one question. You said that you will work on epizoohides in liver stage and in the blood stage, but do you think you can send some Leophilisa or sample of your thing to Toulouse for gametocytes?
I wish we could. It's a funny story. Toulouse are my friends, my colleagues, and they are also like me IRD researcher, but they are not really easy in working with Maison Artemisia because they say, oh, we're going to be attacked because we work with them. And I said, this is really, really silly. We have perfect relationship. We have all the perfect honesty, trust, non-intervention. It's just a perfect relationship. But they're afraid that their paper will be rejected or criticized. I've been asking them that we need to be collaborating because this is silly. We're part of the same institute. We're French researchers, so it's slowly changing. But they have also very strong personalities, and the fact that we got this INR funding is a bit big. Like they feel they will be absorbed and they want to keep their independence. But I agree on that gametocyte assay is very interesting. And to give maybe a very quick word, if you're on clinical trials, in my opinion, I know it's very important that we go there. But if we do clinical trials too early, if we take Pamela Weather's plants and she knows them and they're active in vitro and they're consistent and robust and everything, it's really not bad. But if we take plants really in an uncontrolled way, what happens? If we find by chance a very active plant and it cures people, it's great. But then everybody's going to say, they don't have this background. So everybody's going to say, oh, it cures people. And then people will start using any strain and it will not be as active. The other way around, if we choose by mistake, by mischance, a non-active plant, then it's not going to be active for everybody. Nobody will believe that there is any potential. So if we choose bad or plants, either positive or negative results, it will have bad long-term consequences. So for clinical trials, we need to push them, but we need to push them with great caution.
Yeah, so right there.
Thank you for your presentation. As I said this morning, I'm not at all on the Artemisia part, the plant geneticist, and all what you are showing on sampling, what kind of sample, what the best combination is really interesting. Actually, is what we are doing when we are doing any kind of what we call G by E by M, genetic environment and management system. So I don't know. So I would have one question for you and one recommendation. The question is, do you have any idea of the genetic diversity? Is there any genetic studies on the diversity?
I have really no idea.
Yeah.
Because it would be.
I have no idea. On Anua, I think Ian Graham and Pam will probably comment better than I do. Ian Graham in UK has been making knockout strains for synthetic clusters, synthesizing the precursor of artemisinin or knocking down the flavonoid pathways. So they have mutants. So we know the clusters. We can follow the clusters in wild type plants, but it's not a full coverage of the metabolism. And regarding this diversity, Anua can be probably prospected if we need it. Afra, as far as I know, it's really terra incognita. We don't know. We know what it's producing chemically, starting to know a little bit. But from a genetic standpoint, I don't even know that we have the tools to really amplify specific synergy.
You don't need to amplify, just remark us to look at the gene.
Yeah, I would gladly exchange with you because I'm really not an expert.
And the recommendation, I don't know.
Yeah.
I know some colleague of mine working in Sacramento, working on the genetics of wheat or pig crops, and they do this kind of G by E things. They could, if you list their crop, they can find between your plant A, B, and 3.
Yeah, I'd be very happy to. But again, I will probably not get that, pure agronomical control. Like I said, I need to be probably mixing up qualities under cluster form. Like I will not have one homogeneous family of certain samples. So I will try to simplify the picture into smaller problems and have homogeneous clusters as far as I could, as much as I could. Thank you.
So, yeah.
Thank you, Roman, for your presentation. And it seems to me that Artemisia annua has a long way for at least 30 years, 35 years of the strong researches, then result in varieties. And nowadays we harvest one hectare or 10 hectares. You have a very good average dimension, in terms of artemisinin or so on. And you know well the toxicology. In case of Artemisia afra, do you think is a good way to follow the same process to arrive at one varieties to be more stable when in the end, when the standardization of this?
Well, I think those plants are very, very different. One is really from South Africa, Southeast Africa, Afra. Anua is Eurasian, and it's called more Asian Eurasian. So they are spreading because of human propagation. They go outside the gardens, outside the fields. Anua is propagating in the north. It's old story, very old story. Afra is propagating in Africa, from southeast and South Africa. So I don't think the plants are the same. I really think that they have really different stories to tell us. The fact that one doesn't have Artemisia is also very interesting. I don't think because of that history, maybe they are at the same page, genetically speaking. Like if Anua has been associated to treating malaria for 3,000 years, like poppy was propagated by humans, like cannabis was propagated by humans, like salad were propagated by humans from wild bitter salads. We have now lettuce. So I think the history may be responsible for also a different evolution. Maybe Anua already evolved due to human mass selection. Maybe wild type Anuas, wild type Anuas are not that big. Maybe they are smaller, like cannabis federalis. I think small trees of cannabis. And Anua and Afra is just starting to go to different countries. It's starting its own human association. So I don't think they can be compared. And Afra, as far as I would say, the fact that it's much less known, we should also treat the plant with a little more caution and do more biological assays. And it's probably very strong, but I suspect there might be more toxicity in certain strains, maybe not all. But we know that for hepatic stages, for instance, we had some background hepatotoxicity in vitro. And it needs to be really looked again and see if it's a consistent trait of Afra, or is it just one bad strain and then we should exclude it.
It's finished? No, last question. One question. Last. Yes.
So this is more of a great plan. I like this plan. I can't wait to see the results. So toxicology is interesting because, I mean, there is a fair amount published, certainly for Anua. There is some published for Afra. We have noticed and I'll show some of the data, I guess it's tomorrow. In cell cultures, you see more toxicity with Afra than Anua. But in vivo, both of them are extremely non-toxic in rodent studies with looking at their LD50s. So it's really kind of interesting in terms of that. And it'll be interesting to see if there's any differences amongst these plants in terms of toxicology.
Maybe if you have like metabolization, this is something we partly see on hepatic stage assays because we have sometimes primary hepatocytes, so they cannot reflect of what's happening in the liver. But when you treat cells that don't have detoxification, maybe compounds are very toxic, and maybe in the mice they would be maybe eliminated or turned to non-toxic compounds or active compounds.
Right. You're familiar with that article we published a couple years ago on looking at the mechanism of effect on a couple of P450s in the liver with Afra
probably not. No,
and
no.
Yeah, I'll be glad to share
Yes,
it with you.
please do.
Be
Yeah.
useful.
Awesome. Thank you. Bye.
Thank you.